Biddy explains meiosis in pictures. The purpose of the activity is to improve your understanding of meiosis by illustrating the stages below, using various materials. These materials must be real items around your house. You must take real pictures, taken with your phone or a camera. No digital representations or drawings allowed. There's 8 stages, 8 pictures that you need to take. The structures that you need to represent with items are the centrioles, the nuclear membrane, chromosomes, 2 different colors, cell membrane, and spindle fibers. I have a legend here that explains, it's a small legend, but you can make it bigger, that shows you an example. So there's little starry things that were used for the centrioles, and the rest of the things were different color yarns in this specific assignment. So you create a legend, look to the right, then you arrange each stage of meiosis with your own items. After each stage is arranged, take a picture, photograph the model. The picture must include the stage name, be taken in bird's eye view, and you must arrange your photos in the stages of meiosis in order. So here's how to take a picture. Bird's eye view. See how my phone is looking straight down, minimum shadows. So make sure there's some light there. You can see there's a little bit too much shadow there. So make sure that you have a good picture being taken. Make sure that the identity of each structure is clear in the photo and consistent from stage to stage. Don't change centrioles for one into the other, because I need to see that you understand how they move and work within the stages. You do not have to label the structures, but you must make a legend for those structures for the purpose of clarity. You have to label the stages. Okay? Practice moving the cell structures through each stage of meiosis before taking any photographs. This way, you can avoid having to redo any early stages if you decide that you need to make changes in the materials. The assessment is on Brightspace in the rubric attached to the assignment. So I'm going to show you a couple of assignments from last year. Last year we used mitosis. This year I realized that we need to do meiosis because that is what's in the curriculum. So I kind of messed up last year. There is mitosis in the curriculum, but there's more focus on meiosis. So here's a great one that I found from last year. The stages are really small, so please make the pictures bigger so I can see them better. There is the stages there with the names on the top. Another one here. This is a great legend with all the stuff being used. And then each picture, see how big it is? Each picture shows each phase. Now remember, you have to do 8 pictures. This is similar to what your first stage is going to look like. All right, I'll show you a couple more. This one was a great one. I like how the Q-tips were bent for anaphase and cut in half for telophase. Next one. This one was a good one as well. The pictures are really clear. The pictures are small but very clear. So if you can make them a little bit bigger, that'd be awesome. Remember, this is mitosis. You're doing meiosis. And the last one here, great one here as well. See how there's cleavage furrow happening in telophase as well. All right, so I hope that helps. And if you need any more help, just let me know. Thanks.